Cloning. Cloning is a process used to create a genetically identical copy of a living organism. So far, scientists have successfully cloned DNA, cells, tissues, and whole plants and animals. A lot of commercial plants are created by a process called plant tissue culture, where part of a plant is cut off and a new plant is grown from the cutting. But it doesn't stop at plants. Scientists in the UK made history when they cloned Dolly the sheep. They extracted DNA from an adult sheep cell and inserted it into an unfertilized egg, which they had removed the nucleus from. The egg was implanted into a female sheep to grow, and Dolly the sheep was born. Cloned animals may have some benefits. They could be used in medicine for testing new drugs and medical treatments. They may also be useful in agriculture, as clones of animals that produce a lot of meat or milk. Cloning could also be used to save certain animal species from extinction. Nevertheless, the cloning process is so expensive that none of these options are very practical at the moment. Cloning has not yet been perfected, and many cloned animal embryos do not survive. Dolly the sheep was the only clone to be born after 276 attempts. Cloned animals also often have defects, such as large organs and problems with their immune systems. They also age fast and die young. Dolly only lived for 6 years, half the average lifespan of a normal sheep. As for cloning endangered species, that idea is not a good one, because producing a population of genetically identical organisms would only mean that they would lack the genetic variation necessary for species survival.